Thank you, Andy, and thank you everyone for attending this session and for giving me the opportunity to show you the work that we have done at the University of Illinois with the assembly of 9 insect genomes. During my talk, I'm going to particularly focus on the contributions of the TELSIC libraries to these assemblies. During my talk, I am first going to give you an overview of the DNA sequencing facility at the University of Illinois to put my talk in perspective and so that you understand why am I talking to this about this. Then I will talk about the relevance of this work involving the assembly of insect genomes, and then we'll go over methods, results, and conclusions. Regarding the DNA sequencing facility, we are a service facility. We are 12 research specialists dedicated to user support, to DNA and RNA quality control, to making libraries and doing sequencing, troubleshooting, and offering further support. So we are pure service facility. We have all the latest instrumentation for next generation sequencing, including 2 NovaSeq. These instruments give us a massive sequencing capacity for anything that needs, either has a very large number of samples or needs a very high sequencing depth, such as genome resequencing, RNA-seq or differential gene expression, methylation, exome sequencing, or any other application that needs very high depth. Not all projects need millions of reads, so for those projects such as sequencing of small bacterial genomes or viral genomes or amplicons, we use 3MySeq. For long read sequencing, we have a PacBio Sequel2e, and we also have an Oxford Nanopore GridION. And we also have quite a bit of robotic automation for preparation of library prep in a high throughput manner. So what type of work do we do with all these instruments? We serve the needs of all the University of Illinois campuses, but many of our users are also from academic institutions, private companies, and federal agencies across the United States. And actually, a significant portion of our funding comes from researchers from other countries. And we have users in South America, in Europe, in Middle East and in Asia. So this wide user base means that we get to process all types of samples from numerous plants, animals, microbial communities, and microbes. As a result, a significant portion of the work that we do is in non-model species. Many of these species have no reference genome, and we are contacted to perform de novo genome assembly and for building the genomic resources for all the applications that come later, such as genome resequencing, differential gene expression, and others. So we have a lot of experience in de novo genome assembly. The goal of this particular project is to contribute to the Earth Biogenome Project. So the Earth Biogenome Project aims to sequence all the eukaryotic species on Earth. And so the idea is to contribute to this project by sequencing all the insect species from Illinois. And you may not think of Illinois as, the Amazon, as a source of, enormous source of biodiversity. However, in Illinois, there are over 3,500 insect species. So we have plenty of diversity in insects. The researchers that developed the vision for this work and received the funding for the pilot project are Gene Robinson, Chris Dietrich, and Jeffrey Stein. They are professors at the University of Illinois. All the bioinformatics work that I'm going to show and a lot more that was done to get really great assemblies that I'm not going to have time to show was done by Chris Fields, Kim Walden, and Gloria Rendon from our biotech center. Kim Walden particularly has over a decade of experience doing de novo assembly on insect genomes, so her expertise was critical for this project. And my lab was responsible for DNA extraction, library prep, and sequencing. So why insects? Insects are over half of all the eukaryotic species. So it's a really rich phylum. It's a very diverse phylum. The insects involve, either pests or disease vectors or pollinators or insects that actually recycle nutrients. So they are really important in our ecosystem. The insects used in this study were captured in central Illinois. And you can see that we captured insects from different ecosystems, and they were captured by vacuuming, by sweeping, and also using lights at night to capture nocturnal insects. The insects were brought back to the lab alive. They were classified, that they were put in vials and immediately frozen at -80. We did some preliminary studies showing that the best way to get high molecular weight DNA was to freeze them right away after capture. Then some duplicated insects were added to the collection. So the species that were selected for this particular project represent 6 different orders, 8 families. Some of them are predators or parasitoids or herbivores. But the important thing is that none of them had any genome information. So some of them have very little DNA sequencing, but no genome information previously available. These are the insects that were selected for this project. We call them EB1 to EB31. So this is Earth Biogenome 1 to 31. Here is the order, here is the family. And here you can see pictures of these insects. You can see, one of the things you can see, these pictures are not at scale. So some of these insects are really tiny, such as this one and EB27, and some others are really large. But one thing you can see is that the diversity on the amount of DNA that we will have available per insect, and also the amount of pigments. These pigments sometimes stay in the DNA, and pigments can inhibit downstream reactions and can really affect the efficiency of sequencing. So it's extremely important to develop a method that really cleans up the DNA so that there are no problems downstream. So this diversity of morphologies and pigmentation presents a lot of challenges. In addition to this, insect genomes are particularly difficult to assemble. For example, closely related species may have widely different genome sizes and features. What you learn from one species may not apply to a closely related species, because they may have a completely different number of transposons. They may have transposon insertions from horizontal gene transfer. They may have long telomeres or incorporation of mitochondrial genes into the nuclear genome, or many differences. So you can never guess the genome size of an insect just looking at the genome size of a closely related species. Another thing that is very variable, so is of course the repeat content is highly variable among different taxa. And the other thing is heterozygosity. These are wild-caught individuals, and insect genomes are known for having very high heterozygosity. So a lot of challenges here. And now that we have insects, we need to extract the DNA. And here I can show you an overview of the methods that we use. First, we extracted DNA. And the idea is, because of the heterozygosity, we wanted to do the most we could for the majority of the libraries from only one insect. So we extracted DNA, then we made the TELSIC libraries. TELSIC libraries were used to estimate the genome size and also for scaffolding of the PacBio assembly. Then we made a PacBio library, either Hi-Fi, ultra-low, or CLR. And then we also made Hi-C libraries. So we bought the OmniC kit from Dovetail Genomics. They trained us how to make the libraries ourselves in our lab. And these are the Hi-C libraries that we used for scaffolding. So the assemblies were based on PacBio sequences. We made a PacBio assembly. Then we scaffolded the PacBio assembly with TELSIC libraries, and then we further scaffolded it with the Hi-C libraries. Once we had a final assembly, we did the gap filling with the TELSIC, using the TELSIC reads, and there was a lot of QC done on the final assembly. And we also extracted biological content, also using the TELSIC libraries. So regarding the DNA extraction, let's go to DNA extraction now. If you want to use long reads, it is critical that the DNA is in long fragments. So ideally, you would have the DNA in fragments over 40 kb. Another important thing is that the DNA must be clean. So the absorbance ratios should be 1.8 to 2. We see when the absorbance ratios are 1.2 or even lower, that DNA does not perform well. It really affects the efficiency of sequencing. For PacBio libraries, you need quite a bit of DNA. You need 1 to 5 micrograms depending on the genome size and on how many SMART cells you want to sequence. If the genomes are really small, so less than 500 megabases, then we can start a library with 5 nanograms of DNA. We can do an amplification of the DNA. So for those, we need very little DNA, but the genome must be shorter than 500 megabases. For TELSIC libraries, one major advantage is that you need very little DNA. So depending on the genome size, you will use 1 to 5 nanograms of DNA. Specimen preservation was critical. So if we need to get high molecular weight DNA, the DNA has to be in long fragments. And we saw that if we put the insects in ethanol, for example, ethanol fragments the DNA and nicks the DNA, we didn't have good results. If we tried to put the insects on DNA stabilization solutions, the DNA was also fragmented. The only thing that really worked was to put the insects immediately, either on dry ice or in the freezer at minus 80 alive. In a specimen, and I'm going to talk about this later in the presentation, specimens with large body sizes, it's really important to use only the head and torso and avoid the abdomen. This is something that you can only do if the specimen is really large. So regarding the method, it was a very simple method. We submerged a metal rack in liquid nitrogen. Then we did grinding of the specimen. We put the one insect in a 1.5 ml tube, and then we grind the insect with a disposable plastic vessel. We added CTAB, incubated for some time. Then we added protein SK and RNase, and we incubated overnight. We saw that this overnight incubation was really important to get cleaner DNA. Next day, we clean up with chloroform and isoamyl alcohol. We run, and then we run it through the MAGATRAP kit. This is the kit that we use for most of our high molecular weight DNA extraction. It's bead-based, and we see that it really keeps high molecular weight DNA, and we can wash out several contaminants during the bead washing. Extremely important during the entire procedure is to use white bore pipette tips and to never vortex the DNA. So for example, when we do the cleanup with chloroform, we just rock the tube from side to side very gently. There's never a vortexing step. You have to maintain high molecular weight DNA. How do we evaluate the integrity of the DNA? So you can either run them on a fragment analyzer or a femtopulse. When we did the study early this year, we didn't have a femtopulse. The femtopulse is more sensitive. It can evaluate more high molecular weight DNA. But we had a fragment analyzer, and the fragment analyzer can show clearly, for example, in this sample, that most of the DNA fragments are over 40 kb. So they are all 40 kb. There's minimal fragmentation, basically almost nothing under 40 kb. So this is excellent high molecular weight DNA. Here the peak is anywhere from 80 to 200 kb fragments. In this other sample, for example, we see that there is some high molecular weight DNA, but there is also a lot of fragmentation down to 1 kb. And one thing that we observe is that, that when the DNA is fragmented, it's also nicked, and nicking is fatal for single molecule sequencing. So in this DNA, for example, there would be no point in trying to get long reads and using DNA that is completely fragmented. This is not a DNA that we would use. We would go back to the lab and extract a new DNA. In the insects, there was some variation in DNA quality. We had to accept in some insects some fragmentation down to 10 kb, but nothing under 10 kb. So now, this is the amount of DNA that we got from the different individuals. You can see that we got, for some of them, we got a few micrograms of DNA, so 3.6 micrograms, 6 micrograms. From EB14, remember EB14, because we will go back to this 2 or 3 times in the presentation, we got only 300 nanograms. Here we got quite a bit of DNA. And from these two, EB27 and EB29, we got 18 nanograms. and 6 nanograms. These were really tiny insects. And from this other one, 1.7 nanograms. So what is important is that we wanted to make the TELSIC libraries and the PacBio libraries from one individual. We did not want to mix individuals because then we had to deal with heterozygosity during the assembly. So once we had the DNA, the next step is to make the TELSIC libraries, which we use for estimation of genome size and scaffolding of the PacBio assembly. The TELSIC libraries are linked long reads that are sequenced with short reads. And you can see here there was a paper published in which they explain in detail how the libraries worked, and they show several examples where they assemble microbial genomes and human genomes. So I encourage you to go to that paper and learn more about the libraries. And basically, it's important to start with fragments at least 20 kb. The reason for that, you have to think that PacBio will give you reads that are 10 to 20 kb. So if you want the TELSIC libraries to make a significant contribution for the scaffolding, the DNA should be longer than that. So the DNA has to be as long as possible. The next step is to insert transposons to each fragment. And here, for example, you have here the pictorial view. Here you have a fragment of DNA, let's say it is 40 kb. And then we add transposons in a ratio depending on the genome size and the amount of DNA that we have. We add transposons so that the transposons will end up being spaced several hundreds of base pairs apart, anywhere from 100 to 300 or 400 base pairs apart. So each fragment will have several transposons. The next step is to add the TEL beads. And the TEL beads is a bead that has hundreds of thousands of oligos. And these oligos have a barcode. So each bead has one barcode sequence, has thousands of oligos, but one barcode sequence. And the transposons anneal to this barcode. So each fragment of DNA will go to one TEL bead. One TEL bead can capture anywhere from 4 to 8 different fragments. But because DNA is super coiled, you expect that the entire DNA fragment will anneal to one bead. So the next step is to add another transposon that will fragment the DNA in between two transposons. And then because this transposon, the green transposon and the red transposons have different, let's say, oligo sequences, then we can do a PCR amplification that will amplify this DNA fragment in between the transposons. So now, and then we sequence the library in the Illumina NovaSeq. So now what we have, and this is what you have, the way you have to think about it, is that we started with an entire genome, but now we beam this genome into millions of baskets. Now what we have to do, instead of assembling the entire genome, we just have to assemble these fragments, these 20, 40 kb fragments, 60 kb fragments, using the barcode information. So first we assemble these long fragments, and then we can do the genome assembly using the long linked reads. Of course, if that 40 kb fragment has regions of long repeats or very low diversity, this is based on short reads. So some of them may not assemble well, but the idea is that many of them will assemble entirely. So now we have, so regarding the method used to make the TELSIC libraries, the important thing the take-home message is that they are really easy to make. What you do is you put 1 to 5 nanograms of DNA in a tube, then you add a reagent, then you add the TEL beads, and then you add another reagent to the same tube. That takes about 1 hour. Then you wash the beads, then you add the reagents for the PCR amplification, you do the PCR, and then you clean up the libraries. That's it. So it's all done in one tube, and then you QC the libraries. So it's all done in one tube. It is quick and easy. There is no mystery about it. You don't need any special equipment. You can train anyone, in minutes. Regarding the amount of DNA, that will depend on the genome size. For example, a small genome, 100 megabases, you would use 1 nanogram. A human-sized genome, you would use 3 to 5 nanograms. You have to dilute it to 1 nanogram per microliter accurately. That's where we spend most of our time trying to get that DNA to 1 nanogram per microliter. Sometimes we have 1.3 nanograms per microliter. We dilute it a little bit, and we go to 0.8 and so on and so forth. So it's very important to to start with a very accurate dilution of the DNA. Once we make the library, we run it in the fragment analyzer, and this is the profile that we get. So most fragments are anywhere from, library fragments are anywhere from 300 to 1 KB. Once you remove the adapter, so this means that the fragments are 100 to 400 bases or so. This is how the library looks like. This is the structure of the library. So you have the P5 adapter. This is what adheres to the flow cell. This is the library index. So if you have different samples, each sample will have a different index. So you can pull them all and sequence them on one lane in the NovaSeq. So sequencing starts with read one, and read one starts with a fragment, with the DNA fragment. Then the second read will be the DNA barcode. This is the TEL-seq barcode. So this barcode tells which fragment this belongs to. Then you can reconstruct that original fragment by looking at other sequences that have the same barcode. So you do read one, which is the DNA. Then you read the barcode. Then you read the library index, so you know which sample you are sequencing, and then you sequence read two, so the same DNA fragment from the other end. And for those of you that know how to run the multiplexing pipeline, this is how you put it in the machine. You say, I want a read one, I want another read that has the barcode, I want the index for the multiplexing, and I want read two. So the really important thing to see in this picture is that read one and read two are shotgun reads. They don't have any other information that you have to know or remove. They are simply shotgun reads. So you can make link reads when you use the barcode read, but the read one and read two are just shotguns. So what are TEL-seq libraries used for? They are used for estimation of genome size and genome features. We will see that in my presentation. They are used for scaffolding of an existing assembly. They can be used also for gap filling of a scaffolding assembly. We did that. You can also use them by themselves for de novo assembly with a Supernova software that was developed for the 10x Genomics libraries. Those of you that have been doing de novo assembly know that there were very similar libraries from 10x Genomics that were discontinued. And the TEL-seq libraries took over. They are very similar. They work in a similar way. And you can use the same software to do de novo assembly with these libraries. And because they are shotgun reads, you can also use them for error correction of Oxford nanopore reads and PacBio CLR. So two types of reads that have a high error rate and need to be error corrected. You can use the TEL-seq reads. The sequencing depth is you need to sequence to about a 15x of the haploid genome for estimation of genome size, 30x for scaffolding, and if you want to do de novo assembly with the libraries themselves, then you need to sequence them to 100x. So how did we do the estimation of genome size? So we made the libraries, sequence the libraries, then we input the read one and read two into this software Jellyfish. Jellyfish will count the k-mers in read one and read two, and we generate a histogram of a k-mer distribution. And then we load this histogram into GenomeScope, which is a website where you can just enter the histogram, and it will give you this curve. So this is EB2029. This was a haploid male. Here, for example, the Jellyfish first takes the read one and read two, and it will break them up into substrings of 21 bases, and it counts the number of times that each string is present. So here you have the distribution that you get from k-mer counting, nice distribution. Here you have a lot of k-mers that are very abundant, but they are present only once. And if they are present only once, chances are they are coming from sequencing errors. So this is usually sequencing errors. These are just the k-mers present in the genome. And with this information, the software can calculate the genome size. So here, for example, this is a genome about 134 megabases. And you can see here that it has a very low level of heterozygosity. This is EB29, which is okay. It shows two humps. And what you can see here is these are k-mers that are present in only one of the chromosomes. They're observed and only one, so in only one of the chromosomes. Here, there are k-mers that are present in both chromosomes. So what you are seeing here is heterozygosity. So here, for example, this is a genome size. It calculated a genome size of about 275 megabases. And you can see that the heterozygosity is much higher than in this one, because this one was a haploid, so there was no heterozygosity. So what this shows you is the importance of using only one insect for sequencing, sequencing the genome. This heterozygosity is normal in insects, so we don't see anything weird here. If we see other bumps in here, that could be due to maybe contaminants, or could be due to very long repeats or very abundant repeats or something like that. But there is nothing that, red flags in this genome. However, here we have EB14, which we call the ugly. And the ugly, you can see that there is no curve. Here, something happened that the software was not able to converge and produce a curve. You can see that the estimate of genome size was 1.7 to 3.7, so a very wide range, something strange. And heterozygosity also doesn't make sense. So when we see this, it can still be assembled, of course, if you throw a lot of libraries and money into it. But the cost of assembling this genome was outside of the scope of the pilot project, so we dropped it from the project. However, I will show later, we did an assembly with the TEL-seq reads alone, and we got very interesting data that explains why we were not able to get a good fit in this genome. So now we have to make, so this is the estimation of genome sizes that we got from the EB1 to EB31. So genome sizes were 360 megabases, 1.6 gigabase. Here EB14, we dropped it, so so it doesn't really matter. But the others were 1 gigabase, and these two, you remember that these two were really tiny insects that we only got a few nanograms of DNA. Well, unfortunately, the genome sizes are very small, and so we can still sequence this. So we got the TEL-seq libraries, we calculated the genome sizes. Now we can determine, okay, how much sequencing do we need to do? Now we can budget. We can see how much, how many libraries we need to make and how many SMART cells we need to sequence. So now we go to the PacBio libraries. And we made 3 types of PacBio libraries. The first were Hi-Fi. Hi-Fi libraries need a minimum of 1 microgram of DNA. The DNA has to be long because you want long reads. And they are sequenced to a minimum depth of 20x, and each fragment is sequenced several times. So it produces, and every time it will correct the errors from the previous pass. So it produces error-corrected data, and it really takes a few hours to assemble. Then the ultra-low, remember those 2 insects that had only a few nanograms of DNA, but the genome sizes were very small. So we can prepare these ultra-low libraries. We only need 5 nanograms of DNA, and we had 5 nanograms in those both insects. And the genome sizes were very small. It only works for small genomes. And the reads, because it's based on amplification, on PCR amplification, the reads are usually anywhere from 5 kb to 12 kb. And they are sequenced as Hi-Fi, so the reads are also error-corrected. They are ready for assembly. In one of them had an estimated genome size of 1.7 to 3.2 gigabases, and we decided to make a CLR library. CLR libraries have much longer fragments, but the reads are not error-corrected. So you have to do error correction, and we can do that with TEL-seq. So it's a different type of library. So based on the information that we had, we know the DNA yields that we have the genome, estimated genome size. Now we budgeted, okay, what type of libraries are we going to make, and how many SMART cells are we going to sequence from each one. We have the TEL-seq, we have the PacBio. Now we go to the OmniC libraries, and the OmniC libraries are is a technique that cross-links the chromatin in the tissue. So you don't extract DNA. You just use tissue. You cross-link the chromatin, and you keep the 3D structure of the chromatin. So it introduces bonds that freeze interactions between regions in the same chromosome. And these interactions, they might be a few thousands of bases apart, or maybe millions of bases apart. So it can give you really very long-range information about the genome. Then you cut the DNA with endonucleases. This OmniC, they use sequence-independent endonucleases. So it's is an improvement of previous high-C libraries. And then you perform a ligation that favors fragments that were interacting versus fragments that were not cross-linked. So the thing with these libraries is that you need 20 milligrams of tissue for high library complexity. And ideally, you would use one specimen to avoid heterozygosity. If you have small insects, you need to collect several insects from the same location, hoping that they come from the same family or the same colony. But then you are going to have to deal with heterozygosity later. So we were able to make only C libraries from most of them. This one, EB29, was so small that there was no way we could make libraries. Now, for most of the insects, we only had one more individual. And for many of them, we did not have 20 milligrams. So for many of them, we made libraries without the ideal conditions. So if the OmniC library didn't work well, it's not because the OmniC library didn't work. It's because we didn't have the ideal conditions to prepare them. But we wanted to go ahead anyway as a proof of concept of how many insects do we need, what are the ideal conditions to produce a good assembly. So now we have the TEL-seq, we have the PacBio, we have the OmniC, we sequence everything, and now we have to assemble them. So the assembly started with the PacBio Hi-Fi libraries. We did a raw assembly using Hi-Fi ASM. Then the next step was porching duplicates and porching haplotypes. And then we did scaffolding with the TEL-seq rates using links and arcs. Next step is to do scaffolding using ARIMA and SALSA with the OmniC libraries. And then there is a lot of QC that is done. First, we did gap filling with the TEL-seq rates using Sealer, and then block tools and repeat masker and mitofinder. This was very important because looking at the mitochondrial genome, we were able to reclassify some of the insects. So a lot of QC was done that I won't have time to show in this presentation. So now let's look at the results of the sequencing and the assembly. First of all, our first question was, what are the genome sizes? And here we have in blue the actual genome size. When we did the assembly, this is the actual genome size. And in orange, they estimate the genome size from the TEL-seq libraries when we put them in Jellyfish and GenomeScope. The gray is another software that we use, but let's just focus on blue and orange. You can see here it was right in the spot. Here it was a little bit off, but nothing too bad. EB14, it was off, and you will see why. I mean, this is the one that we couldn't assemble. We didn't even try to assemble. EB15 was very close, 17 very close, 19 very close. EB20 was the CLR library, and it was a little bit off, but that has more to do with having used a CLR library. And EB27, 29, and 31 were very close. So the majority of them, the assemblies that work well, the estimation of genome size was very close to the actual genome size. That was a really great finding. And here, this is the assembly metrics. So here we have some of the most important data. I know this table has a lot of numbers, but let me walk you through it. So here we have the insects, EB1, EB5, EB15. Here we have the numbers from the PacBio assembly. Then we added the TEL-seq and for scaffolding, and then we added the OmniC. Here we have the number of scaffolds, the total size of scaffolds, which is the genome size, longest scaffold, shortest scaffold, and this is the most important one, the N50 scaffold length. So in EB1, for example, you can see when we did the assembly with PacBio, we got scaffold lengths of 8 megabases. In general, anyway, anything that is over 1 megabase, we are really happy. But this one was a very good assembly to start with. And then when we added the TEL-seq, we almost got a twice as long scaffold length. So the TEL-seq worked really well for that. The OmniC made a small contribution, but remember, this is not the fault of the OmniC. It's the fault that we made the libraries with only one insect, and many times we did not have the amount of tissue that is needed to make these libraries. EB5, from the beginning, we had an excellent assembly, so the TEL-seq and the OmniC made little contributions. And also some of the TELSIC libraries, for some of them, we did have some fragmentation of the DNA. So some of them, they didn't have a lot of fragments over 20 kb. So so that's something also to take into account. EB15, for example, the TELSIC libraries duplicated the size of the scaffolds, and the OmniC libraries made a huge increase in scaffold length 50 and also on the longest scaffold. EB17 is another one that made a significant, the TELSIC library made a significant contribution, and the OmniC library added more, longer scaffold and much longer. The longest scaffold was also much longer. EB19, again, the TELSIC library produced almost 3 times as long N50 scaffold, and the OmniC library produced this is very likely a chromosome. So it made a significant contribution. Now I'm going to show the same table, but I'm going to add one column. So don't get scared by the numbers. I added one column here, the Busco scores. We compare our assembly against a set of almost 1,400 conserved genes that are supposed to be in most eukaryotes. And we can see here, what we can see here is that 98% of these genes were present in our assembly. In most cases, they were present in single copy, duplications were very low, and in most cases, the genes were complete. They had no fragmentation. So, so this means that our assemblies have a really great assembly of the gene space. All these genes that we were looking for are there, they are in single copy, and they are not fragmented. So, and that also means, the point I'm trying to make here is that when we added the TELSIC libraries and then the OmniC library, we did not break the gene space. So that was for 5 of the insects. This is the other 4 insects. EB20 is the insect where we used CLR libraries. And you remember that the estimated genome size was 1.7 to 3.2. It ended up being lower, and the assembly is definitely not that good. It's lots of scaffolds, and the scaffold length 50 is not really high. It took an enormous amount of time computationally to get the assembly because of the high error rate and because of the error correction. So what we learned here is next time, if we have a genome size estimated between 1.7 and 3.2, we will do Hi-Fi. Hi-Fi works, and CLR is extremely computationally intensive, and it didn't work that well. EB27 was another one in which the TELSIC libraries made a major contribution, and the OmniC libraries added more to the assembly. EB29 the insect was so small that we could not make an OmniC library. But you can see here that the TELSIC library made a major contribution, so 8 times longer scaffold length, and the longest scaffold was much longer also. So that when all you have is a TELSIC, then that's the way to go. And EB31 is another one in which both the TELSIC and the OmniC libraries made significant contributions. And again, I'm going to show the same table with the Busco scores, and you can see the CLR, we are missing several genes in there, but the others are really great assembly of the gene space with no fragmentation. So this is regarding the assembly metric. Now let's look at some of the QC of the assembly. And here, for example, we have EB5. Here, what we do is we blasted the scaffolds to the UniRevPro database for classification, of tags. What is in our assembly, and we also aligned the TELSIC reads to look for statistical significance. And we can see here, for example, that all the scaffolds, basically the majority of the scaffolds, blast to arthropode. So the dark red is arthropode. And so this is really, the core insect genome is all arthropode. And here there is an outlier that aligns to bacteria. And this is very likely an endosymbiont. This is very common in insects to have endosymbionts. And with the TELSIC libraries, we can see that the reads are in sufficient depth that this is real. This is not just a spurious artifact. This is real. This exists. So we were able to identify an endosymbiont. That's really great. Now you remember the ugly. In the ugly, what we did not make any PacBio or Hi-C library, but we did assembly of the genome using Supernova. It took forever. It took over 3 weeks, and we got, of course, a really bad assembly, very fragmented assembly. And the reason for that is this, that when we align the scaffolds to the database, we get here in dark red are again arthropod, but about 1/3 or maybe almost one half of the reads are something else. And this something else is bacteria, fungi, plant, and nematodes. So this is a herbivore, and it's feeding on the plant and whatever else is on the plant, and so everything is showing. It's a very small insect. Remember, it was only 300 nanograms, so it was very difficult to, if we remove the abdomen, then we end up with almost, no DNA, and this has a long genome size. So we can, with this information, we can go back and we can remove the scaffolds that belong to these bacteria and plants, and we can also, using Kraken, we can remove the raw reads that align to bacteria and fungi, but there are still many reads that are unclassified and that still, make the assembly difficult. So this would be an assembly that it probably can be done, but you have to throw a lot of resources into it. And also you will have to use many insects because the insect is very small. And that will further complicate the assembly because then you have to deal with heterozygosity. We also wanted, we do that after doing scaffolding, we do that, to put two contexts together, and there will be a gap in the middle. And so we wanted to fill that gap with the Telsig reads. We can use the Telsig reads for those. And I wanted to see the Telsig reads, what's the coverage of Telsig reads along the genome. And this is the EB29 genome. We align the Telsig reads to the genome, and we also align the raw hi-fi reads. And you can see that the entire genome is represented by Telsig reads. There's no portion that is missing. When we look at a small portion of this alignment, you can see here, peaks and valleys. This is very typical of libraries that come from PCR amplification. There are some biases, but at the end of the day, the entire portion is covered by reads. So we can use Telsig reads for gap filling, and we can also use them for error correction of a nanopore assembly or a CLR assembly. Final considerations. Regarding the pilot project, we validated how insects could be preserved. We validated a method for high molecular weight DNA extraction. We validated construction of Telsig libraries, PacBio and OmniC libraries. We also validated the workflow for genome assembly and QC. And remember, there was a lot more QC than what I am showing. We saw that by getting the information from genome size and features, that really helped us to determine the sequencing depth and the effort expected for each genome. We learned that next time we will only use hi-fi libraries. We will not use CLR libraries because they really took a lot more time to assemble that one than all the other genomes. And we didn't get a good assembly. We know that we have to capture as many insects from the same area as possible, mostly for OmniC libraries. And then if you have a small insect with a large genome, you will need a lot of insects. And whenever possible, we know that we have to avoid using the abdomen. Regarding specifically the Telsig libraries, we know that they require minimal. First of all, the major advantage is that they require minimal amounts of DNA. So we can make libraries from the same individual that is used to make the PacBio libraries. So we don't have to deal with heterozygosity there. So the same individuals will do libraries and scaffolding. It's a very easy protocol. You don't need any special equipment, no microfluidic equipment. It has a very low cost. The cost in reagents per library is $100 to $200, depending on the genome size, on how much DNA you have to use. The sequencing depth is low. If you can put a lot of libraries in one S4 lane, so the cost of sequencing per sample is low. The evaluation of the genome size and genome features was very important for us because it really allowed us to determine how much budget and effort we have to put into assembly. It's the only scaffolding tool available when the amount of DNA or tissue is limited. That was the case in EB29. That's all we had for scaffolding, and it produced a significant improvement of the assembly. As you can show, as I show in the data, there's many times when they are done in the right conditions, they produce significant improvements in F50 scaffold sizes. And also they allow for gap filling, so gap filling and also error correction with highly accurate reads. So compared to reads with high error rates. And they can also be used for de novo assembly by themselves if no other long reads are available. And my last slide is another example from a plant. We also assembled melon, but in melon we did something different. We made TEL-seq libraries and we did PacBio libraries. And we assembled the TEL-seq by themselves using Supernova. The melon genome is about 440 megabases. And you can see with TEL-seq we assembled almost the entire genome. And the scaffold length was 3.5 megabases, so it was very decent. Of course, with PacBio, we got the entire genome, much longer scaffolds. And when we scaffolded the PacBio assembly with TEL-seq, we got much longer assemblies. But if for some reason the only thing we had available was a little bit of DNA, we could have made the assembly with TEL-seq really quick, really easy, and we could have obtained a decent assembly. And with that, I wanted to thank, first of all, the PIs, for their vision, the vision they have for the entire project, sequencing all the insects in Illinois. My lab for the amazing work that they do for making the libraries and doing the sequencing and everything they do, caring so much about quality. The bioinformatics team and also the team that maintains all our sequencing computing infrastructure. The Office of the Vice Chancellor for Research, because they support our biotech center. And the funding for this pilot project came from the Illinois Innovation Network. So thank you for the money. And now I'll be very happy to answer questions.
Thank you, Alvaro. As a reminder to webinar participants, if you have a question, please type it into the Q&A box. And if you haven't already, please take a moment to fill out our survey questions now by clicking on the green survey icon at the bottom of your screen. For the Q&A portion of the webinar, we will be joined by Tom Chen, PhD, Chief Scientific Officer at Universal Sequencing. The first question is for Alvaro and is about some of your samples. With some of the insects, especially the leafhopper or others, would holding them for a period of time to allow clearance of the gut or feeding them something like a defined solution allow clearance and diminish the potential contaminating material in your DNA prep?
That's a great question. And yes, absolutely, yes. That's one thing that can be done. Some of the insects, unless they are feeding, they die right away. So it's you really have to know about the biology of the insects, in which ones you can starve for a while before you freeze them. But yeah, that's something that we're going to look at, and yeah, it has come up, and absolutely, it can be done in some insects.
How many different parts of a genome will bind to one bead? Is it possible to have parts of more different chromosomes barcoded from the same bead?
So great question. Yes. So each bead, for example, in these large genomes, eukaryotic genomes, each bead will bind anywhere from 4 to 8 genomic fragments. So those 4 to 8 genomic fragments will have the same barcode, but that's okay because the genomes are really large, and the chances that the same two regions in the genome will bind the same bead are very statistically very, very low. So you know that these two fragments will have the same barcode, but they have a different sequence. So they belong in different regions of the genome. The question is also important because previous libraries that were similar to the TEL-seq libraries, they had about 300 fragments that in each barcode bin. And these libraries have only 4 to 8 fragments, so they have an extremely high definition in each one of the bins. But yeah, the answer is about 4 to 6 in eukaryotes. And if you go down to microbes, for example, where really, you have a very tiny genome, then it's just 1 or 2 fragments of DNA that bind the tail bits. And you do that by the dilution and the ratio of DNA to bits when you start the protocol.
How amenable is TELSEQ to mammalian or human samples, including human samples?
Yeah, so for human samples, then the paper that I showed, that the first paper that they published, it shows some assembly of human genomes, and I think most of these long-read linked long-read technologies, they are really great for human, cow, all mammals that have much simpler genomes. I think that they are a really great alternative for mammalian genomes. In plants, when you go to plants and you start dealing with polyploidy and you start dealing with extremely long repeats or something, then they are still perfectly fine to use for scaffolding, but to use them only for de novo assembly, you might run into problems. But I think for easy genomes, as mammalian genomes, they can be even used for assembly just by themselves.
What is the largest genome that you attempted to do a de novo assembly for using only TELSEQ libraries?
No, only TELSEQ libraries was that one, the ugly. But it was it was an extremely complicated genome that had so many other things that it was never going to assemble. But other than the melon that was 400 megabases, we haven't, because it's not our objective. Our objective is if we have plenty of DNA, we will do PacBio and then use the TELSEQ, the strength of the TELSEQ for the estimation of genome size, for the scaffolding, and for the gap filling.
Can TELSEQ libraries be sequenced with 2 by 250 reads, and is there an advantage of using longer reads?
So they definitely can. For example, if you are going to use it for de novo assembly, Supernova, I believe that it only takes 2 by 250 reads. So even if you import the 2 by 250, it will clean them to 2 by 150. However, for scaffolding, all those, the arcs and links, for example, they are designed for even for very long reads, for monopore or PacBio reads. So yes, definitely you can use 2 by 250, and there could be some advantages. Any region of low diversity or something, having longer reads will be an advantage. The reason why we haven't tried it is that we want to keep sequencing costs as low as possible. And when you look at sequencing 2 by 150 in the NovaSeq and sequencing 2 by 250, the 2 by 250 is comparatively much more expensive than the 2 by 150. So we have only done 2 by 150, but I think for scaffolding, definitely you can do 2 by 250.
How does TELSEQ work for sequencing mitochondrial DNA, and is it possible to reconstruct mitochondria from the DNA isolates?
I would probably give that question to Tom, because we haven't gone to such small genome. I suppose if you can really purify mitochondrial genome without any nuclear DNA, I don't know if you can with a TELSEQ go to such low genome, about 16 kb or so.
Sure. Let me chip in. Again, we not intentionally to sequence mitochondrial DNA or sequence directly, but based on the overall the whole genome sequencing, we do see there's good coverage on the mitochondrial DNA, and it's in principle it depends on what's your purpose. For sure, we know it's the mitochondrial DNA is covered. However, if you have a way can specifically identify and isolate the mitochondrial DNA, we definitely also can sequence. But overall, so far, we only at the whole genome level, we do see there's a pretty good coverage on the mitochondrial DNA.
Yeah, and also in our assemblies, for example, we were able to identify, which in most of them, which of the scaffolds were from mitochondria, and based on that, we were able to reclassify one of the insects. We thought it was one species, but based on the mitochondria, we had to reclassify it. So if you sequence whole genome, whole genome DNA, you will get also the mitochondrial genome.
This is another question for Alvaro. What was the total sequencing cost per insect?
Yeah, so for this pilot project, we spent anywhere from some sequencing cost plus bioinformatics and everything. It was between $3,000 to $5,000 per insect. Yeah, that's what it was.
Can you use TEL-seq reads?
I think you got cut off. I didn't.
Yeah.
I'll say it again. Can you use TEL-seq reads to polish an assembly?
So, for example, in the assemblies that we had that were based on PacBio, the assemblies are phased. So if you use shotgun reads to polish the assembly, you will remove that phasing. The PacBio reads are already high accuracy. So there is no need to polish a PacBio assembly with short reads. You can definitely use the TEL-seq, and that's what we did for filling the gaps, right, in between the contigs. You can definitely use the TEL-seq to polish an assembly that is based on Oxford Nanopore and on PacBio CLR. So that's a major advantage. You know, you make one type of library, just the TEL-seq, to do scaffolding, to do polishing, and to do gap filling. But a PacBio hi-fi library, you don't want to polish with short reads. You can polish with long reads, with the reads themselves, but not with short reads.
Do you think TEL-seq could be used in combination with target enrichment sequencing?
I would have to know what's the what's behind that question. I don't think I follow it completely.
Yes. So maybe let me chip in. Yes, we do have the procedure for targeted sequencing. That depends which way you refer to. So we have a way so we can enrich non-contiguous target region. For example, is over 20 kb, even 100 kb. Then because usually the enrichment yields will be low, that will actually perfectly fit into the TEL-seq procedure. We can use as low as 0.1 nanogram to making the TEL-seq library for the targeted sequencing.
One of our audience members notes that some core facilities may be hesitant to try a new sequencing primer or may not have enough clients to fill a whole flow cell for TEL-seq, and asks, is it offered as a service?
Definitely, yes, absolutely. We have done, we do de novo assembly for people from all over the world. And in many of our assemblies, we have included TEL-seq. So we offer the entire service. And if you just want us to make the TEL-seq libraries, we can also just make those. As long as the DNA really meets the requirements and is clean and is long, and we can make the libraries.
All right. Well, that's all the time we have for today. We'd like to thank Alvaro Hernandez, Tom Chen, and our sponsor, Illumina. If we didn't have time to get to your question, we will try to follow up with our expert. As a reminder, please look for the survey after you log out to provide your feedback.
Thank you. Thank you very much for listening.
Thank you.